In this lecture, we're going to talk about the organelles. Organelles are small structures that are found inside of our cells, and organelles, when they are working, then the cell is working. Some of these organelles are membrane-bound organelles. And so membrane-bound organelles include the Golgi apparatus, the endoplasmic reticulum, lysosomes, peroxisomes, and the mitochondria. So let's look at their jobs, what they look like, and how they function. The endoplasmic reticulum is a series of tubes that move from the nucleus all the way out towards the Golgi apparatus. And so it's like a network of tubules that runs outwards from the Golgi, from the nucleus towards the Golgi. The endoplasmic reticulum has two forms. So you have either rough endoplasmic reticulum, which contains ribosomes, or smooth endoplasmic reticulum, which does not contain ribosomes. So here is your nucleus, and here surrounding the nucleus is the rough endoplasmic reticulum. And then coming off the rough endoplasmic reticulum, we have smooth endoplasmic reticulum. And then we'll have the Golgi coming out here as well. So the rough endoplasmic reticulum functions in processing proteins. So protein production occurs on the ribosomes in the rough ER, and then the proteins can be modified. Carbohydrate group might be added, or you might just add a heme moiety or another moiety to the group. And then once it is formed, then we just surround it by a vesicle, so a membrane sac, and we send it from the rough ER to the Golgi. The smooth ER does not contain ribosomes, so there's no protein production here. Instead, we're producing molecules like lipids, cholesterol, estrogens. The smooth ER also functions in as a holding site for calcium ions. It functions in helping to detoxify the cell from different alcohols and other poisons. So we find a lot of smooth ER in areas where you're going to need extra detoxification, like the liver. The Golgi apparatus then is kind of, I like to think of it as the post office. I know your textbook calls it the warehouse, but I like to call it the post office. A warehouse is good too, because warehouses are where you package things up and then you send them out. The post office is where you package things up and then you send things out. And so the Golgi, you're going to finish modifying the protein, package it up properly, sort it, so you're going to choose where it has to go, and then you're going to send it out. There are three mechanisms for sending the proteins out. And this is just a little more information. Modification, packaging, and sorting occur from the cis phase, and then they move molecules towards the trans phase of the Golgi. So the cis is close to the endoplasmic reticulum, and the trans is farthest away from the endoplasmic reticulum. Here we have different mechanisms for transport. One of these mechanisms is in the Golgi on the trans phase, the blebbing of the vesicle from the Golgi, moving it towards the plasma membrane. The vesicle binds to the plasma membrane, and via exocytosis, we release the materials out. Another mechanism would be the blebbing of the vesicle, but the proteins, instead of being just set in the vesicle, they are embedded in the walls of the vesicle. And so when the vesicle binds to the plasma membrane and open, the proteins become part of that plasma membrane. The third mechanism is the formation of vesicles that stay inside of the cell. So here we have the blebbing of a vesicle, and that vesicle stays inside the cell and contains proteins. In this case, these are lysosomes, so that contains digestive enzymes. And let's talk about lysosomes. So lysosomes are tiny little vesicles that are formed by the Golgi. So the enzymes are produced, and then so the proteins are produced, they move into the Golgi. The Golgi then forms the vesicle that surrounds the enzymes. And so we now have this wonderful membrane-bound vesicle filled with digestive enzymes that can break down other molecules. So it's really important when you have either cells that are dying that you need to degrade, or you're trying to kill off disease. Peroxisomes are membrane-bound vesicles as well. They're similar to lysosomes, but peroxisomes actually form from the blebbing off of a vesicle in the ER, so rough endoplasmic reticulum. And peroxisomes contain oxidative enzymes. When they bind to another molecule, they release these oxidative enzymes that cause damage to the cell. And in the process, they produce hydrogen peroxide, which is also dangerous to cells. But we can take the hydrogen peroxide, and we have special enzymes, catalase, that will break the hydrogen peroxide down to water and oxygen, which is not dangerous. Peroxisomes function in breaking down different molecules through oxidation. One of the examples is beta oxidation of fatty acids. So remember back in chapter 3, which was just like a little bit ago, since we are in only chapter 4, we talked about beta oxidation of fatty acids, where you're cleaving off an acetyl group from the fatty acid chains. This process occurs by the peroxisomes. The mitochondria is a membrane-bound organelle that contains enzymes for producing ATP. So this is the energy producer, the powerhouse of the cell, and it functions in producing the majority of the ATP. This is something we talked about in chapter 3. So another thing that you should already know, because we just talked about it. And almost all of our cells have mitochondria for energy production. Now we'll focus on the non-membrane bound organelles. The first is the ribosome. Ribosomes are structures that, what they are, they're RNA and proteins that are formed in the nucleolus of the cell. They're released to either sit in the cytoplasm, or they embed themselves into the endoplasmic reticulum, forming the rough ER. Their function is to produce proteins, which we'll talk about a little later in this chapter. So ribosomes function to produce proteins. That's their job. Ribosomes have two subunits, a large subunit and a small subunit. Together they form the functional ribosome. A large subunit of the ribosome is called a 70S ribosome. The small subunit is called the large subunit is called a 60S subunit. The small subunit is called a 40S subunit. The full functional ribosome is called an 80S ribosome. And that's important because when you get into other courses like microbiology, you'll learn that ribosomes help us to determine differences within species. And it helps us so that we can target different diseases. We can attack the ribosomes of other organisms because our ribosomes differ. The cytoskeleton. The cytoskeleton is an important structure that's composed of microtubules, intermediate filaments, and actin filaments. It functions in maintaining the cell integrity, so it's bound to the plasma membrane and helps to maintain the cell structure. It also functions in moving around organelles and other materials, and really importantly, it functions in cell division, so moving DNA around so that our cells divide properly. Centrosomes are formed from the microtubules, which microtubules are one of the largest, actually, of the proteins that make up the cytoskeleton. And so centrosomes are formed from microtubules, and they contain, they're formed from microtubules, and they function in producing spindle fibers. The spindle fibers come out from the centrioles, which are found within the centrosome, and they attach to DNA. And then they can move the DNA along the cell and pull it from one side of the cell or the other, and so they control mitosis. That's really important because we need to have identical copies of DNA when our cells divide, unless we're producing sex cells, which is meiosis, but then we need half the amount of DNA, so it's still important. Cilia and flagella, these are structures that are found on the plasma membrane. They actually project out from the plasma membrane. Cilia are found on certain cell surfaces, and their function is to move materials along the cell. So we have ciliated cells in our bronchi, in our trachea, and what these ciliated cells do is they move mucus and material, so mucus that gets trapped, no, not mucus that gets trapped, mucus that has trapped material like, say, dust from the air, and they move it away from our lungs so we don't take in that stuff, and then it gets to our throat where we can either spit or swallow that down. So it protects us. We have cilia in our why can't I think of the word? Fallopian tubes, our uterine tubes. These cilia beat towards the uterus, and they move the egg that we have produced towards that uterus to hopefully be fertilized and then embed in the uterus. Flagella, and I like this picture better. This shows the cilia really nice, okay? Flagella are similar in structure to cilia, but they typically have, well, actually, in the human, they only have one tail unless there's some type of a mutation occurring, and they function to move the entire cell. The only human cell that is flagellated is a sperm cell. This is just kind of a cute joke, so don't be offended. Here's an egg, and it says, "How gentlemen died." "After you, sir," says one sperm. No, after you," says another sperm. "Get out of the way, you jerks," says another sperm who's running towards the egg. Ha ha, just a joke. The last structure we'll talk about then are microvilli. These are also extensions from the plasma membrane, but they're very, very small. They're microscopic. Cilia are microscopic, so they're all microscopic, but these are super tiny, and they are really, really densely packed typically. They don't function in movement. What they function to do is increase surface area for absorption. So the more surface area we have, the better we can absorb materials. We have microvilli in areas where we need a lot of surface area for absorption. So think about, where do you think surface area for absorption is really important? No, I'm not going to get an answer since I'm talking to really you, but you're not here. I'm talking to myself, or like my little Maid of the Mist duck or something, right? That's sitting here with me. So one place is the small intestines, where we're absorbing nutrients from the foods we eat. Another place is the kidneys and our nephrons, because we need to reabsorb all the nutrients that were filtered out of our blood. So in this section, I'm going to finish up talking about some structures that help to either hold our cells together or help our cells to communicate with each other. So cells come together and they form what we call tissues. For cells to be able to come together and be held together, we have to have something that holds them together. So we have these structures called junctions that hold cells together and that hold cells to underlying tissues. We also have some junctions that just allow for cells to communicate between each other. Oh, you know what? I want to talk about this first one. The first one is called a tight junction. So we see tight junctions in between cells, and they help to hold the cells together so that nothing can leak through the cell. Okay. We have desmosomes, which are right here, and hemidesmosomes that hold cells together. They connect cells together, and they protect the cells from tearing, wear and tear, from mechanical stress. Hemidesmosomes anchor cells to underlying tissue. So the tissue that lies beneath the epidermis, the dermal tissue, is called connective tissue. And then we have gap junctions. Gap junctions are junctions that have tiny little pores, so you can see them right here, and they allow for communication. So nutrients can pass from one cell to the other cell, or electrical impulses can move from one cell to the other cell. So we basically can speak from one cell to another cell. And here you can see the tight junctions, the desmosomes, the hemidesmosomes, and the gap junctions. We will stop there, and in our next video we'll talk about the nucleus. The nucleus is really important, and there's a lot of stuff that goes along with the nucleus, including, but not limited to, haha, right, protein synthesis and replication of DNA, so cell replication. So replication of DNA allows for cell replication. And so we'll talk about that as well. Have a great evening or day, whatever time it is for you. Bye.